Good morning, students. We are going to continue chapter 6 of NCERT, that is Molecular Basis of Inheritance. Students, till now we have discussed the replication of the DNA, transcription of the DNA, and translation of mRNA. Now it's time to discuss more about the DNA, how the genes are going to be regulated according to the requirement of the cell. This is being carried by the strand of the DNA, and that particular strand of the DNA which is going to control whole of the gene expression altogether is referred as Operon. So whole of the video is going to be devoted to the single topic, that is gene regulation. So let's begin. First of all, we need to understand what gene regulation actually means to you. It means that controlling the amount and time of formation of the gene product according to the requirement of the cell. As you know, the gene product are required according to the requirement of the particular cell. So this is going to be controlled according to the amount and the time of formation of the gene product. In eukaryote, gene regulation means controlling the amount and time of formation of the gene product according to the requirement of the cell. As you know, in eukaryotes, the growth is highly regulated and it is being in concordance with the expression of the gene at a particular time with the amount. Okay, so in eukaryote, gene regulation happens at four primary levels. That is at the transcription level, processing level, transport of mRNA from nucleus to the cytoplasm, and at the translational level. How it is being carried out at the transcription level? The regulation of the primary transcript being formed. What kind of transcript is being used for the process for the formation of the mRNA? Processing level, the regulation of the primary script formation, that is very important. How the capping and tailing process is going to be conducted along with the splicing process, because there are so many introns which need to be spliced out from the final mRNA. And then the transport of the mRNA by the nuclear pore, it actually happens. At the transport level, there is also the regulation of the gene expression, because the mRNA which is being required by the cell is only going out of the nucleus, otherwise it is going to be stopped there. Okay, the last level is the translational level. We have discussed at the translational level there are UTRs, so that UTRs are actually going to control the gene expression altogether. Now it's time to discuss about lac operon. Before going to the deep detail of the lac operon, first of all we need to understand what operon actually means. It is a concept which is being proposed by Jacob and Monod. Okay, this name, the two name, that is Jacob and Monod, are very important because they have proposed the concept of Operon. Operon is a unit of prokaryotic gene, because it was first being explained in the prokaryote. So that's why it is being referred as prokaryotic gene expression, which includes the coordinated, regulated structural genes and the control which are recognized by the regulatory gene product. Because there are several genes in the DNA which are actually going to regulate or control the amount of the gene product being formed at a particular time. So there are so many regulatory gene sequences being present in the prokaryotic DNA, which is polycistronic in nature. So the prokaryotic gene always have the regulatory sequence being attached, so that whole of the process of formation of the gene product is going to be controlled according to the time and amount being required by the cell specific. So, first of all, we need to understand the lac operon with the help of the diagram. Here you can see the promoter, two promoter side. PP is the promoter, I is the inhibitor, and O is the operator gene. You can see the repressor mRNA is going to be formed, and ultimately with the repressor mRNA there is formation of repressor protein which is going to be bind at the operator region, and this repressor ultimately blocks the entry of some of the enzymes which are being required for the synthesis of mRNA. One enzyme you know, that is RNA polymerase. So RNA polymerase entry is going to be blocked over here. So the rest of the gene are not going to be transcribed at all. But when inducer is going to be present, the inducer is going to be bind with the repressor, and ultimately that repressor is not going to bind at the operator region. So this site is free, and RNA polymerase can join over here, so whole of the process of transcript formation can take place. That's how the regulatory gene sequence is going to control the expression of rest of the gene. That is I gene, that is inhibitor gene, is going to be control the rest of the gene transcription. So this I gene is referred as regulatory sequence. Now this lac operon actually being described in E. coli. Okay, this is a E. coli gene regulation, and you know the food material for the E. coli is the lactose. Whenever lactose is going to be present in the medium, so these genes are going to be transcribed. When lactose is going to be absent in the medium, so these gene product are not being required, because these genes z.ya are the genes which actually help in digestion of the lactose by the E. coli. But when the lactose is absent in the outer medium or the surrounding medium or the culture medium, then there is no point to synthesize mRNA which is able to metabolize the lactose. Okay, so if this process is going to be continue in the absence of the lactose in the outer medium, then it is a kind of wastage for the whole of the cell in terms of energy. And energy is very very precious in case of prokaryotes because they have limited stuff to synthesize the energy. That's why they need to conserve the energy, and this energy is going to be conserved when repressor is going to be bind at a particular site, so that whole of the metabolite gene are going to be stopped or halted, and their transcription is going to be stopped altogether. So it is a more like controlling the gene expression in terms to save the energy for the cell. And now comes to the function of z.ya enzyme, that is referred sometime what I used to refer, that is z.ya. Z.ya Z is going to synthesize beta-galactocidase, Y is going to synthesize permease, and A is going to synthesize transacetylase. Beta-galactocidase is the enzyme which is going to convert lactose into galactose plus glucose. Permease induces the permeability and causes the changes into that of the cell membrane of the E. coli, so that more and more lactose can be allowed inside that of the cell. And the last enzyme is the transacetylase, which is being synthesized by the A gene. And transacetylase actually activate the lactose in the form of L-lactose. Acetyl group is going to be attached to the lactose and it becomes L-lactose, and L-lactose is ultimately going to help as that of the inducer. Okay, so that's how the gene regulation is being controlled in E. coli. And we have various type of Operon. This Operon we are studying, that is lac Operon, that is lactose Operon, and so many Allerectron Operon or trip Operon. All sort of Operons are going to be present. So we are going to discuss this particular Operon in great detail in this presentation. Components of an operon. We have seen the structural gene, promoter site, operator site, regulatory gene, inducer. So these are the five components of the operon. Structural gene, the fragment of the DNA which transcribed mRNA for the polypeptide synthesis. Structural gene, that is z.ya, we have discussed. That is a gene which is actually being required by the cell for the various metabolic functions of the cell. The promoter site, the sequence of the DNA where RNA polymerase binds actually, it is referred as promoter site. As you know, the transcription start with the enzyme that is RNA polymerase. And this RNA polymerase here is DNA dependent, so the full form will be DNA dependent RNA polymerase. Operator, the sequence of DNA adjacent to the promoter where specific repressor protein binds is called that of the operator. And regulatory gene, as we have seen I gene, the gene that codes for the repressor protein that binds to the operator and suppresses its activity, as a result of which transcription is going to be switched on. After the production of the repressor protein, the repressor protein is going to bind with that of the operator region, and operator region is blocked by the repressor protein, and repressor protein is going to be synthesized by the regulator gene. And then we have inducer. Inducer is a kind of substrate that prevents the repressor from binding to the operator, is called an inducer. As a result, transcription is switched on. It is a chemical of diverse nature like metabolite, hormone, substrate, etc. It can be of any type of thing like metabolite, as we have seen in the case of E. coli, that is lactose. Okay, so hormone can also be an inducer sometimes when the growth of eukaryotes is going to be concerned. Now you can see over here, this is a beautiful animation. Here you can find out the promoter region and two promoter region. They are being demarcated by the repressor, and here that is a promoter region is being demarcated with that of the lac, that is lactose operon. Okay, so here is a repressor gene, and repressor gene is going to form repressor protein, and that repressor protein is going to be bind with that of the operator region. Okay, you can see here the operator region is going to be blocked by the repressor protein, and repressor protein when bind with that of the operator region, then whole of the synthesis of z.ya is going to be stopped. Z.ya is going to be stopped. Okay, so that's how this actually work in the negative control, because repressor protein is going to block the transcription of z.ya. When lactose is going to be present into the outer medium, and it is being absorbed by the E. coli, that E. coli ultimately led to the binding of the inducer with that of the repressor protein. When repressor is going to be attached with that of inducer, then this sequence is free to bind with that of the uh, RNA polymerase, and RNA polymerase then continue the synthesis of the mRNA, and mRNA synthesis will lead to the formation of the polypeptide chain, that is enzymatic sequence are going to be formed. So this is the animation which actually talks about how gene expression is going to be controlled into the E. coli in terms of metabolite being present outside of the cell. When there is a food, the E. coli is going to synthesize the enzymes which are able to digest that particular kind of food. If there is no food, there is no usage of these enzymes, so whole process is going to be halted down by the repressor protein. Now comes to the lactose operon, like Z Y and LAC-A genes are transcribed from the leg transcription unit under the control of single promoter. We have seen one promoter is there because it is a polycistronic gene. They encode enzyme required for the use of lactose as a carbon source. Okay. The LAC-I gene product, the LAC repressor, is expressed from the separate transcription unit which is being present at the upstream. That is prior to that of uh, promoter gene of the z.ya. The repressor gene will be there. Okay, so upstream means before. Now comes to the lac Operon unit. You can see three genes, that is Z are going to be controlled by the promoter region, and there is also one operator region which helps the binding of the RNA polymerase. If RNA polymerase is unable to bind, why it is going not able to bind? Because there is presence of the repressor protein. The three structural gene, that is Z, transcribe the polycistronic mRNA, and Z is encode beta-galactocidase. Beta-galactocidase role is to break lactose into galactose plus glucose, and gene Y code for the permease. Permease is that kind of enzyme which actually causes the alteration into the cell membrane of the E. coli. Then it's going to allow more and more lactose come inside of the cell. So it increases the permeability of the cell membrane to absorb more and more lactose from the outer medium. The third enzyme, the last enzyme, which is very, very important, gene A codes for the enzyme, that is transacetylase, which catalyzes the transacetylation of the lactose in its active form, that is allolactose. Now come to the part what happened to the whole gene when lactose is absent, and what happened to the gene when lactose is present. So we have to study two concepts. When lactose is absent, we are discussing first part, that is regulatory part. When lactose is going to be absent, I gene regulates and produces a repressor protein, and that repressor protein is going to be bind with that of the operator region, and that region is very crucial for the binding of the RNA polymerase, and that going to be blocked by the repressor protein. Then how RNA polymerase is going to bind at that particular site? That particular site is totally being blocked by the repressor protein. Okay, so there will be no transcription of the Z.ya will take place. The Operon is switched off. So whole of the system, the formation of the genes, that is transcription of the genes, will be stopped. Okay, when lactose is present. When lactose is present in the outer medium, then uh, that lactose is going to bind with that of repressor protein. Then there is no site of the repressor protein which can bind with that of the operator. Operator is totally free to bind with that of the RNA polymerase. Then the transcription of the Z.ya will continue. Okay, so the lac Operon is going to be switched on. From this point of time, you can easily see there is a both positive and negative control. So we can say it is totally a negative kind of control, but it is both kind, that is positive and negative. If lactose is present, it is a kind of positive control. If lactose is going to be absent, it is a kind of negative control. So both control mechanism are there in the lac Operon. Okay. This is the summarized diagram of whole of the process. You can see the regulatory sequence in the upstream region, and z.ya, z.ya is there, and there is operator sequence. Here you can see when there is no lactose is being present in the outer medium, the repressor transcript is going to be formed, and that repressor transcript ultimately translated into the repressor protein. That repressor protein is going to bind with the operator region, and that operator region ultimately stop the transcription of Z.ya. When lactose is going to be present into the outer medium, then this repressor protein is going to be bind with that of the inducer, that is lactose or allolactose. This is going to be inactivated. Then DNA dependent RNA polymerase can move into the downstream for the transcription of the Z.ya, and ultimately led to the formation of the enzymes which can digest lactose and increases the permeability of the membrane to absorb more and more lactose from the outer medium. This how the gene expression is going to be controlled. I hope you have done whole of this slide, and this is very crucial from the examination point of view. Operon has been asked two times since 2010 in the CBSE board examination as a 5 marker. So you can understand the relevance of this topic from the CBSE point of view. You have to go through whole of the slide very well, at least twice, so that you can understand the concept in a holistic manner. And please, students, makes note. Notes are going to help you out at the end movement revision, which is very crucial for the board examination. That's all for today. Thank you.